Good morning to you all present over here. Last time we seen questions regarding with a nucleosome number. Okay. Today we will see that how this, because central dogma we seen last means the last lecture. Inside that what we seen that a DNA replication it forwarded to transcription means formation of mRNA. Then it forwarded to formation of protein or polypeptide synthesis. So this unidirectional flow of information, it is known as what? Central dogma. We also seen reverse transcription or taminism. Okay. So further we have to see that if we are seeing that differences between this DNA and RNA, so why this DNA is more stable and why RNA is not considered as genetic material. Okay, genetic material is that, but not proceeding in as replication. But what happens? This RNA is what? It is evolved first. So RNA is the genetic material that develops first, and from that, because that at that time it was unstable, and due to stability, this RNA converted to DNA, and the stability that provides DNA, it is the essential genetic material that being used everywhere. Okay, so we have to see that the properties of genetic material regarding with DNA versus RNA. So come on that. DNA versus RNA. Now, before going into that, we have to know that RNA was the first genetic material, and many evidences also proved that RNA was the first genetic material. So, what I have to take? RNA was the first genetic material. Okay, this RNA was the first genetic material, and many evidences also proved that RNA evolves first. Okay, but why the problem happens? But due to unstable in nature, unstable in nature, it not acts as genetic material except some viruses. Okay, now DNA acts as genetic material which develops from RNA due to its stable nature. And how this stability come? Since DNA contains thymine, which provides stability to DNA. Stability to DNA, whereas RNA contain uracil, which makes unstable. Okay, so now you understood that RNA is what? It is genetic material, first genetic material. that gives rise to DNA that was stable. But if we are comparing this reacting on the basis of reactivity, so RNA is most reactive but unstable. DNA is less reactive but stable. So what we can say here? Now, what we have to say here, that any genetic material must have capability of replication. Okay, so DNA have the capacity of replication. Next, structurally and chemically stability should be there. So what I told, DNA has the capacity of structurally and chemically provide stability, while RNA had more reactivity capacity, reactivity capacity, but less stable capacity. Okay, so DNA has the capacity what? Structurally and chemically provide stability, and that's why it is used as genetic material. Now, DNA also having the slow scope of movement. What I told, slow scope of movement means changes. So DNA had scope of slow changes. That means it also help in evolution. Okay, so that is having which, that is slow changes. Okay, that changes slow, and that's why it helps in what? Evolution. Now, DNA also having the capacity to express the Mendelian characters. So DNA also having the capacity to express Mendelian characters. Okay, so this is the more important, that's why it differentiating from RNA to DNA. Okay, now we also know that why it happens. So RNA is being unstable. One more reason is what? The free 2OH of RNA. So the free 2OH group of RNA becomes it more reactive and less stable, and that's why RNA is not used generally as genetic material. Okay, now also we seen RNA contain uracil that makes less stable compared to DNA that having thymine. Okay, now this entire information showing that RNA evolves first and DNA later. Okay. Also, it shows DNA is used as genetic material compared to RNA. Why? because of its stable nature. Okay, now next we have to come on the central dogma, and inside that first process is DNA replication. So we have to see what are the processes involved in DNA replication. So come on that. It is also known as DNA duplication. So DNA replication or DNA duplication. Now, before going into that, we know that DNA making their copy itself, that is known as DNA replication. So what we are telling? Formation of new DNA from old DNA. Formation of new DNA from old DNA is called formation of new daughter DNA from old parent DNA is called DNA replication or DNA duplication. Okay. Now, next we have to know that in which time this DNA replication occurs during cell division. So we know that already we studied in 11th in cell division. Meiosis is there, okay, mitosis is there, interphase is there. So in the interphase, one phase is S phase, synthetic phase. Okay, so in which time S phase of cell cycle DNA replication takes place, and S phase also full form is synthesis phase. So in which time this takes place? So DNA replication takes place at S phase of cell cycle. Okay, this is very, very important. Okay, medical question, that at which time DNA replication takes place. So what is that? At S phase of cell cycle. Now, another thing is asking that what are the enzyme important? Okay, which enzyme having the importance in DNA replication? So which enzyme is there? Here enzyme name is DNA dependent DNA polymerase. This is also important. DNA dependent DNA polymerase enzyme have important role in DNA replication. Which enzyme is that? DNA dependent DNA polymerase enzyme. Okay. Now, what is the direction that form DNA replication? Means new daughter DNA that is forming. What is that direction? So direction is direction of new daughter DNA formation. New daughter DNA formation is 5' to 3'. Okay, it is what? 5 prime to 3 prime, and on which strand it is forming? So on template strand 3 prime to 5 prime, means on the template strand 3 prime to prime 5 prime, the formation is going on 5 prime to 3 prime or 5 dash to 3 dash direction. Okay, now we know that we initially we seen that, okay, we studied that DNA replication is what? Semi conservative, means one parental DNA and one daughter DNA is being formed. So what I told, DNA replication is DNA replication is semi conservative. Semi conservative, that we already seen. Three types of replication is there: conservative, dispersive, and semi conservative. So it is what? DNA replication is semi conservative, semi continuous. What is that? Another one? Semi continuous. Semi continuous means one is continuous and one is discontinuous. We will see that while going in that in detail. So it is what? Semi conservative, semi continuous, and another thing is what? Bidirectional, means one direction is going on, but another direction is just opposite, anti parallel, bidirectional. Okay, so semi continuous, anti parallel, bidirectional in nature. This line is important. You have to remember what I told. What are the features? Semi conservative, semi continuous, anti parallel, bidirectional in nature. Two directions is having. Okay, now one scientist were there, that name is Matthew Meselson and Franklin Stahl. He proved, or he shows, that DNA replication is semi-conservative by using N15, heavy isotope of nitrogen. How this is going on, that we have to see. So the experiment conducted by Matthew Meselson and Stahl. Okay, these two scientists were there, Matthew Meselson and Stahl. Full name is Franklin Stahl. So we can write that Franklin Stahl. This name you have to remember. Matthew Meselson and Franklin Stahl. Okay. The experiment conducted by this, by using NH4Cl, and N is what? 15. Nitrogen 15, that is heavy isotope of nitrogen. Please be remember, it is heavy isotope of nitrogen, not the radioactive. Okay, this is only heavy isotope of nitrogen. It is not radioactive. One questions came in the medical exam, that Meselson and Stahl experiment used NH4Cl, okay, medium, And options were there, which one? Heavy isotope of nitrogen, radioactive isotope of radioactive nitrogen, both, none of these. So we have to remember that it is what? Heavy isotope of nitrogen, and this used in E. coli bacteria. So the experiment conducted by Matthew Messelson and Stahl by using this in E. coli bacteria. Okay, and we know bacteria every 20 minutes gets doubled. Okay, so if, just for example, I'm taking here, what happens? E. coli, it is getting double 20 minutes. So if one is there, so it will come 2. If another 20 minutes, we are telling 20 minutes, so what we are telling? 20 plus this 20, so it will come 40 minutes. So in 40 minutes, what will happen? 2 to the 4. Next 60 minutes, so how much it will come? 8. Okay, so this we have to remember, that every 20 minutes it getting doubled. Now from this, Ames has asked question. What the questions were there? We will see that. So we have to just remember this one: bacteria every 20 minutes get doubled. Okay, now what the experiment goes in this Messelson and Stahl, that we have to see now. In the initial, what I told, in E. coli, the grow medium was what? NH4Cl. And where, what was that? N15. N15 was heavy isotope. Okay. So here what he does, first generation he used this one. So it is N15, N15. It was heavy isotope. This is the DNA. Now, then what he told, that this occurs on E. coli. Okay, so next what happens here, that seen that while doing a centrifugation, okay, on a density gradient cesium chloride, this you have to remember what I told in the cesium chloride. So what happens here, this was N15. N15 is heavy isotope. N15 and 14 is hybrid, and N14 N14 is light. So these three type of results came while going into Messelson and Stahl experiment. So what here he seen? Okay, so first one came what? Heavy isotopes. So that heavy isotopes come where? It came at the bottom. So this came in the vitro when they are doing that, done that one in the experiment. So what came? This was came N15 N15 heavy isotope. N15 N15, that was heavy isotope. Now then next generation what came? This, just for example we are doing that, this one was like that and this one was like that. So it is what? This N15 N15, this is N15, this is also N15. Now what I told, this is parental, so new generation, new daughter DNA form, so new daughter DNA form here, this is N14. Same here, this is N14. This is what? First generation. First generation means what we can say? 20 minutes. Okay, because E. coli bacteria every 20 minutes gets doubled. Now, here what came? Here came that proportions came here. It is what? This is hybrid. This is also hybrid. So hybrid came above the bottom side here. So this came here. This was N15, N14. It is completely heavy. Here came hybrid. Understood that one? Now, further what happens? This further after that second generation. So what happens here? We have to see this. You understand, heavy, light, and hybrid. So come on that. This further came here. So what we can say? Second generation. This is first generation 20 minutes. This is second generation. So here 20 plus 20, how much will come? 40 minutes. Now, here what came? We have to see now. One was this, another was this. Same thing here. One was this, another was this. Okay. Here what was that? This was N15, N15. This is N14, N14. Okay. Now again what happens? Density, what I told, cesium chloride density gradient. When this capped in the medium cesium chloride density gradient, and that medium contained what? Normal NH4Cl. This was the medium. Normal nitrogen, heaviest isotope is not there. Only initially heaviest isotope was taken. Now here what happens? This also will come. This is N14. This will come. This is also N14. Here this is come N14. This also came N14. Okay, so this is now here what came here? So one came light and one came hybrid. So here what the configuration came in the tube. Okay, so what came here? One came hybrid, this one. This came hybrid. This is N15, N14. Okay, and another one what came? Another came this one, and this one came what? Light. So this is N14, N14. Okay, so what the things here came? Here both came. One it is light and one it is hybrid. Okay, so like that, if we are going further this one, so how we are going on? One changes into two, this two changes into four, if third generation we are going, so it will come 8. Okay, in first generation questions are asking in the examination what? How much heavy particle are formed, or light formed, or hybrid DNA formed. So in first generation what formed? One hybrid. Okay, in second generation what form? Second generation one hybrid and one light. Okay, you have seen that, one light and one hybrid. If this is first generation, this is second generation. If in the for, how much it will come? So you have to see that. So it is showing that heavy DNA containing medium reduced and a light getting more. And this especially shows that DNA is semi-conservative. Okay. Now, one more scientist came. That name was Taylor. So come on that. This you have to remember. Questions are same coming. This figure will come and they will ask this configuration. So this configuration you have to remember. This one, and this one. Mostly these three are asking. Okay. So we have to remember this one. Now come on. Now, come on next. What I told, one scientist were there. Their name was Teller. So he does what? He provided a semi-conservative mode of chromosome replication in eukaryotes by using T3 TDR. T3 TDR thymidine tritiated thymidine in the roots of Vicia faba. So what I told, this also you have to remember. The scientist name was Taylor. Okay. So Taylor provided semi conservative mode of replication of chromosome by using tritiated tritiated thymidine in the roots of Vicia faba, that is beans, broad beans. Okay. Now here you have to remember what that which has proved. So Taylor by using what? Tritiated thymidine, that is T3TDR. Okay. So tritiated thymidine in the roots of what? Vicia faba, that is broad beans. Okay. Now here one questions came. What the questions is there, that we have to see. Then you will understand clearly. What is that? If a hybrid, question is there, if a hybrid. Now hybrid means what? N15 N14. Here we started from heavy astro, but it is starting from hybrid. So be attentive. So if a hybrid is allowed to replication, is allowed to replication. Okay, it is allowing to replication. How much? Thrice. Thrice. In a medium containing, in a medium containing what they are containing? N14 means normal. But it is hybrid means N15 N14. It is N14. Now, what is the proportion of, question are asking, what is the proportion of, what are the things are we are having? Light, heavy and hybrid. What is the proportion of light, heavy and hybrid? Okay, light, heavy, and hybrid DNA obtained respectively. Obtained respectively. Now, options are there. First option is 7 light. Okay, 7 light, 0 heavy, 1 hybrid. Now, option 2 is there, 4 light, 2 heavy, 4 hybrid. Option 3 are there, 3 light, 3 heavy,